back Thursday. I was gone all spring, our fall break, so I'm working on those and Vital Ones tests. But anyway, we have only 2.5 weeks remaining here. Let's take a look at our calendar. We've got basically today, Thursday, and Tuesday essentially for lecture content. So I think that we'll cram in enough stuff for an exam. This will be like a short exam, probably. Maybe just like 4 pages.
I thought we were doing the lab exam first.
Fine, feel shorted. But
No, I have it.
so today we're going to cover sensory cells and the evolution of eyes.
That's what I have.
And then on Thursday and Tuesday, we will cover basically vertebrate physiology, respiration, osmoregulation, and all that stuff. So that's kind of our plan for finishing out lecture exam 4. Remember lecture exam 4, which is on the 19th, is only the last 2 weeks.
My bad.
So it's not going to be a particular part of the test. And then for our lab exam, we have a lab exam on Thursday the 12th, next Thursday. I think we're pretty much geared up for that. Today we're going to do amphibians and reptiles and birds. On Thursday we'll do mammals, so we'll do a frog section today. Thursday we're going to do mammals, and we'll do the rat section on Thursday. And that will leave us this Tuesday for review. Like, you need to review anything, pull the shark out again, look at the frogs, review your rat pictures. Basically, Tuesday, today will be in addition to lecture content. We'll have a review of the lab.
Oh, yeah.
And then our lab exam Thursday will be the last lab exam. Again, it's not cumulative, it's just on stuff that covers embryology, on oddballs, and that kind of nerves.
I think all of my classes are, thankfully.
Okay. And then I think I mentioned on Tuesday the 17th, this will be getting back your lab exams, a review for the lecture exam, and then I'll have like an extra credit, help me clean the lab up today. And that will only take about 20 minutes. It's not a big deal. So whoever's here to help take stuff up and get things tidied up for the spring, it'll be worth some points there. Okay, I don't know how many, but I'll make it worth your while. All right. So there you go. Any questions? Looking good? You're all seem so. You're still on a turkey high or something. What's going on? Okay. All right. Let's go ahead and get going with our topic for the day, which is sensory cells. And because we did our nervous system last time, what we haven't talked about yet is the sensory system. I'm not really going to go over this in super detail for all of the sensory system, but we're going to focus in on photoreceptors and the evolution of eyes. That's a really good example of sensory cells. But anyway, for sensory integration, we have all kinds of ways an organism can sense their environment. Chemoreceptors are responsible for smell and taste. Taste and smell are basically the same essential function. And with the shark, many of you decipher the shark brain. We know that telencephalon, that front lobe of the shark brain, which is actually turns into the cerebrum later, is the smell receptor or the olfactory. So it's chemoreceptors. Mechanoreceptors would be for touch and hearing. We're going to see how the evolution of the ear kind of goes. The lateral line and the ossicles of the fish turn into basically the eardrums of the amphibian and so on. So we'll see kind of the transition of mechanoreceptors. So hearing is related to touch. It's actually a vibration sensing structure. And then we'll skip photoreceptors here. There is kind of a unique electroreceptor that some fishes have. For example, the sharks have these pores on their nostril area where they can sense voltage, from the nervous system of struggling prey fish. And then you probably have heard about electric eels. Electric eels are these fishes that can actually reverse their electroreceptors. to send out a pulse of electricity. They're using their muscles to actually power electrical discharge. And that's all related to the lateral lines and electroreceptors. I think for our purposes, it'll be fun to kind of focus in on photoreceptors and the evolution of eyesight, because that's a really interesting thing about animals if the vision potential is interesting. So anyway, I want to start talking about animal vision with a protozoan, though, because sight in animals, where's my sight in animals actually starts before animals are animals. There are many protists that have vision structures or light sensing structures that probably are homologous to what's going on with the eyesight. So let's look at the euglena. Remember, euglena was a protozoan that had interesting kind of, looks like a chili pepper kind of, it's got a cell shape, and it has a flagellum. And the interesting thing about euglena was that euglena, if you remember, had chloroplasts in it. So the chloroplasts were these structures that convert light energy into cell energy. They convert light into ATP the cell can use. What a chloroplast uses is a molecule called chlorophyll A. There's two different kinds of chlorophyll, but chlorophyll A is the primary one. And chlorophyll A, if you look at it, it's an interesting a molecule. Let's go ahead and pop it up on Google here. Go ahead and take a look at chlorophyll. So chlorophyll, let's look at the model structure of it. Here we go. So this is what chlorophyll looks like. It is a very large kind of organic, it's called a porphyrin head. It's basically this large organic molecule. And to me, this thing looks like, and this is how I kind of interpret a chlorophyll molecule, is it looks kind of like a big tennis racket. And that's kind of how it works, is we have a, let me see if I can do this here. It has kind of like this big octagonal head, and then it has this fatty acid chain tail here that embeds in the membrane. And so what happens with chlorophyll, so it has this big porphyrin head. I'm just going to draw basically a big ring. Just imagine it's like a tennis racket. And how this works is light comes in and strikes or is absorbed by the head here, and then the kinetic energy of the light, because the photons of light are coming in, really fast, the speed of light obviously, and even though they don't really have a measurable mass, there is energy exerted here, so it is, a conversion of kinetic energy into motion of his head. But basically what happens is this, as a result of the light striking the head will bend over, and as a result, an electron is kind of shot out of that little double bonded oxygen that's kind of connecting that bracket head to the handle. So this is basically how chlorophyll works. It's a light sensitive molecule that's very good at absorbing light, and when the light strikes it kind of knocks the head over, kind of like what do we call it, bumping the tennis racket. And as a result, an electron is excited, and that excited electron is then used to make energy for itself. chloroplasts or chlorophyll works. What's interesting to me is the euglena also had, I think I mentioned when we drew it before, it has a light sensing structure here called a stigma. This stigma is a separate light sensing structure that's associated with the flagellum. So, and I thought this is kind of weird, but why would an organism that has light sensing organelles like chloroplasts have a completely separate light sensing structure as a flagellum? And I think I mentioned that at the time, the euglena are able to lose their chloroplasts, or they can shed their chloroplasts if they don't have light, and that they can operate like a protozoan that's heterotrophic. They can feed on other organic molecules. So that stigma is a light sensing switch at the flagellum. Basically, it turns the flagellum on and off depending on whether light is present. So if light's present, the stigma will turn the flagellum on and it'll basically swim towards the light. The stigma contains a molecule called retinol. And retinol is we also call this vitamin A. You know, retinol is basically, some people use retinol as a beauty supplement, you know, to keep your skin moist or what have you, a skin moisturizer. But retinol is, specifically one form of retinol, is a molecule that is essential for sight. So in your retina, which is what retinol is named after, retinol is the primary photoreceptor. Let me show you what that looks like. And retinol, you probably know it as for skin creams or lotions, etc. But if you look at the structure of retinol, let's go ahead and look at our Wikipedia page. Check it out. It is kind of like a ping pong paddle. You know, I mean, it's the same kind of molecule. It has this fatty acid tail here that's embedded in the membrane. And then it has a little ring, but it's like a tiny little ring. So I would say that retinol is kind of like a ping pong paddle compared to chlorophyll. But it works the same way. Light will strike onto that little paddle, and what happens there's a double bonded oxygen here, just like chlorophyll, is it bends over, and as a result, that little double bonded oxygen there will send an electron out. But in this case, what happens is the electron stimulates a nerve cell. So the retinol doesn't have to be as, I guess, powerful as a chlorophyll. Chlorophyll is responsible for making ATP for the cell. The retinol just has to have enough energy to stimulate a nerve, a sensory nerve, to send the message on to photosynthesis. So I'm going to argue that sight or vision, for one, is closely related to photosynthesis. And just as an aside, there are purple photosynthetic bacteria that use retinol as their photosynthesis molecule. So in certain types of bacteria, retinol is actually used as the photosynthesis driving pigment. But it seems that the use of retinol for light sensing probably happened before animals were animals. And euglena is a good example of a kind of like an eye spot that occurs in a protozoan. Okay? So, that being said, let's go ahead and take a look at kind of how this has changed in animals over time. There are light sensing structures in sponges and in jellyfish. Nigerians, box jelly, for example, have little eye spots. But the eye spots in jellyfish are basically the same as what occurs in the flatworms. So I'm going to go through basically several different types of eyes and how the eyes are formed. And that's kind of how we're going to do our comparison. You can imagine like a big comparison graph here. So let's take a look at the plasmon flatworms. Because that is where we saw kind of like the first appearance of sight structures causing a change in body form. Remember, the bilateral symmetry was the result of cephalization or forming of a head. And the head was formed because these eye spots are basically being processed by a cerebral ganglia. And you did all that for the last exam. So let's take a look at our flatworm and how their eyes are developed. And let's see here, we had eye spots here associated with the cerebral ganglia. So let's take a look at how these are kind of put together, or how the tissues were sort of organized on these organisms. So what's going on here is we have retinol patches, or patches of retinol that occur, concentrated in a certain spot that we call an eye spot. So on the outside we have kind of epithelial cells. So we have kind of our epithelium on the outside. So here is our epithelium. There. Okay, and then below the epithelium there is I guess you'd say it's a retinol layer, but we'll go ahead and just call it a retina. So this is the patch of pigment that is kind of the light sensing structures. And then radiating from this, we have optic nerves. So this is the nerve cells that will basically take that signal and convert it into a nerve signal that's sent to the cerebral ganglia for decisions. So here we have optic nerve. All right, so that's basically what makes up, and this occurs in flatworms. This basic system occurs in jellyfish, cnidarians. There are some kinds of sponges that have patches, even though they don't have nerve cells. But even like our lamprey, we saw in our lamprey there was kind of that third eye that occurred at the diencephalon. Even vertebrates have basically an eye patch that occurs, associated with the brain. So this is a very simple, very common system here. And how I probably named this is what would be called an eye spot. So it's basically just a patch of retina that is exciting some neurons that then go and send the message to the brain or the cerebral ganglia or whatever. Okay, so the disadvantages of an eye spot compared to other eyes is that basically it's just for light dark. All this can do is see light or no light, and it can process movement. So imagine like if you closed your eyelids and looked into the light and then waved your hand in front of your face, you can see that there's movement going on. You really can't see any shapes or images. So this is not suitable for seeing actually themes, shapes, images, or edges or anything like that. But it can detect movement. So these little flatworms, if a predator is coming at them, they can kind of respond or react because they can see movement or motion. Okay, you get the idea? So but basically, essentially it's kind of using the same basic principle as the protozoan does. Where we have some advancement in our vision would be with the segmented worms. The polychaete worms, let me go ahead and get a picture here. Polychaete eyes. Polychaetes, which are the marine, they're these marine worms, actually have very, very sophisticated eyes. Here's a good kind of image or example here. So anyway, you get the idea. Let me pull up some better ones. Okay, so the eyes of polychaete worms. These worms are predators, so it helps them to be able to see not only motion, but actually shapes and distances, etc. So we'll go ahead and draw a little polychaete worm here, kind of a marine clam worm or something like that. Earthworms obviously don't have eyes, but they're burrowed underground. But let's see here. We have Keely on here, so we know this is a polychaete. Okay, so this is Annelida. The Annelida have what amounts to simple eyes, or what are also called ocelli. We'll give you the name here in a second. Let's go ahead and draw sort of how their eyes operate. They take this basic same design and they turn it into kind of a cavity or a pocket. Imagine like this being a balloon and taking your finger and sticking your finger into the balloon so you get a dent. So what we have going on here is like what we have going on here is so it's here's the epithelium on the outside. Same idea as what the flatworm has. So here's our epithelium. Okay, and then the retina is in kind of the same orientation, just behind the epithelium zone here. And we have optic nerve comes from radiate from the backside of it. Nerve. So what's going on here is it's basically the same kind of thing. It's just that the eye is arranged into kind of this pocket. And in primitive examples, it's still open to the ocean, so this is filled with seawater. But what this amounts to, the technical name of this is called a simple eye. The real name is called an ocelli, but you could use simple eye on these and it's fine. But how this thing functions is like what's called a pinhole camera. And there is in photography, there's kind of like, in the olden days of photography, when they first invented cameras were kind of like a box that had a pinhole in it. You actually use a pin to put the thing in there. So here's an example of a pinhole camera. You basically make a box, you put a small hole, usually with like a nail or a pin, and what it does basically is it will project an image onto the back side of the box. And usually you put like a piece of slate film or a page of film back there, and you open up the pinhole, take the picture, and then close the pinhole up. But basically, the pinhole, that little whole opening here, this little pinhole here, projects an image onto the back of the eye pocket there. And so these organisms can actually see shapes. In some cases, they have retina that can differentiate colors. They can have rods and cones in their retina cells. And so anyway, this is an advancement, but it's interesting is it's really just a simple modification, a little pocket of your eye spot and turn it into a simple eye where they can actually see images on that idea there. So this is kind of like the pinhole camera example, where you can, it's really just a simple modification. It really doesn't require that much change in the actual structure of the organism. So ocelli occur in segmented worms. They occur in mollusks, like certain kinds of snails, etc. And then you can also see those in the eye patches on top of the heads of insects as well. So ocelli are a pretty widely used type of eye structure. Okay, then we move on to what would be called more complex eyes. I guess the limitations of ocelli are that even though these organisms can see shape, it's a very nearsighted situation. So their vision probably is like 1 meter. With a simple eye like this, the ability to see maybe 3 feet would be about as max as they can do. So it's a very, very close vision type situation. And the image is not very sharp because it doesn't have any kind of a lens or anything like that. So the simple eye is obviously useful because you can see shapes and movement and colors in some cases. But what are called more complex eyes are for what we consider as vision standards. So let's take a look at the insects or arthropods have what's called a compound eye. And a compound eye, let me go ahead and draw an example of an insect that has a compound eye, like a praying mantis would be a good example. Praying mantis. Some mandibles here. And then the eyes of the praying mantis, I'm using the black to indicate what the eyes are kind of these domes that have individual little facets compounded together or compiled together. So this is compound eye. So here is insects or arthropods. Okay, so let's go ahead and take a look at what this compound eye is made of here. And in this case, what we got going on is the eye is made up of hundreds or even thousands in some cases of individual little eye pockets or facets. So here's kind of how this works. Blue is still epithelium. What we're doing is we're showing kind of how it's arranged together. This. Okay. And then, let's see here. Retina is at kind of the back of the pocket. And then the optic nerve radiates from the back of these structures here. Okay, so here's our optic nerve. Here is the retina, which is kind of the back of these pockets. And then here is the epithelium. Now the insects also have an exoskeleton or a cuticle. And the cuticle basically covers the outside of this and adds an extra layer of structure here. this cuticle provides a bit of a lens, or a kind of a transparent focusing lens onto the structure. So there is kind of like a lens effect of the cuticle, where the image or the light comes in, and it operates, each individual little facet operates like a simple eye. But when you put a whole bunch of them together, what's going on here is they're actually, each facet is seeing an image. This is what the facets kind of look like. But the insect's brain, the insect's cerebral ganglia, are actually able to put this together into a single composite image. So the insect's not actually seeing, separate little facets like this image shows. But what the insect is seeing basically is kind of a composite image that is put together by the brain. So each one of these little facets, this whole thing is called a compound eye. And it is basically like a whole bunch of little simple eyes that are put together into a cluster. And in the case of like praying mantis, it would be, thousands of them kind of all compiled together on each side of the head. Each one of these eye facets can see an image. Again, the image is kind of blurry. It's not a super sharp image, at least far away, but the image is actually good probably for, well, I'm going to say maybe as much as 3 or 4 meters, like 10, 12, 15 feet. So a horsefly, for example, which has probably one of the best eyes of all insects. These are biting flies that look for mammals to feed on. They can see, a possible prey or a possible host for getting blood, from 15, 20 feet away. So these compound eyes basically have sharper vision farther out than a simple eye does. The other advantage of the compound eye is they're super motion sensitive, because each one of these little eye facets is like a separate little eye. When the brain puts it all together, insects are super able to detect motion or movement, because kind of each eye has its own little thing going on. But anyway, when you look at a praying mantis, I like this picture because this whole thing basically is a compound eye. If you see that little black dot there and there, it looks kind of like a little pupil looking at you. But in fact, what's happening is, that's where you're looking down maybe 3 or 4 individual little tubes. So it's kind of like you're looking down little tubes here, and on the rest of these, you're basically looking at the epithelium of the cuticle, which is why you're seeing green. But why you're seeing black here is you're actually looking down into the tube, and that's where the retina is absorbing the light. So that's as you kind of move around or pan around the insect's head, you'll basically be looking down into the tubes. Very interesting. So the compound eye is sophisticated. Again, these insects can see 10, 15 feet away, shapes. But it's not a very, very sharp image. It still has kind of limitations like the simple eye does. Okay? So, moving on to what is considered to be the kind of the best option here for sharp vision is what's called a camera eye. And the camera eye occurs in both vertebrates have a camera eye, and certain kinds of mollusks, namely squid. You'll probably notice that almost every topic here has a squid example. We call this squidology, right? Zoology. But anyway, mollusks have a type of camera eye, and vertebrates have a type of camera eye. So this is like the 2 main organisms or groups that have this camera eye example. Let's take a look at the squid first and show how their camera eye works or functions, and then we'll look at the fish as an example of the vertebrate camera eye. Okay, so here is the mollusca, namely the the cephalopods, the octopus and the squid, have a camera eye. And many of you actually dissected these eyeballs out. And so let's go ahead and take a look here at how these things are put together. Here we go. I'm having a squid looking that way, so I know everything else is going to look the other way. All right, here's our epithelium here. So kind of the same orientation, or so far it looks very similar to a simple eye situation. We have a retina that occurs behind the epithelium. Here's a retina. And then we have an optic nerve that essentially senses what the retina is seeing. So far, this basically is identical to a simple eye, except we have a lens that occurs here on this side. So we have a lens that essentially sits in the opening of the eye and can focus or adjust what the image is on the back of the eye. So the big difference here is the size of the pocket. The pocket here is filled with fluid, that's body fluid, and not seawater. So on the simple eye, basically we had this, seawater was basically filling that area. Here, it's basically body fluid called humor, which is the kind of the fluid in your eye is called the humor of your eye. And so when the squid dives down deep, for example, it doesn't compress or change shape. But what happens here is because it's a larger pocket, maybe a centimeter or two in size, and there's a lens that focuses the light that comes through, is these organisms are able to see much sharper vision. So this is called a camera eye. And basically it is made up of a pocket that the image is projected on the back, the retina of the eye. This is fluid filled, and the lens basically can focus or adjust that image as needed. So that's basically, and I think those of you that dissected this out, when you dissected this out, you weren't able to actually pull the whole eyeball out. It wasn't really an eyeball, it was an eye pocket. What you pulled out was that lens that was sitting there right in the opening. And I think many of you got that thing out. It's kind of a round, like thing. I know I'm drawing flat here, but it's kind of more of a round structure there. So that's what, one version of a camera eye here is for the mollusca. What's super interesting is in the vertebrates, fishes, for example, we saw in the shark, they had an eye also. Here's kind of our fish example. So here's the eye of a fish or a shark or whatever. And let's go ahead and take a look at how this works. Is the fish has a camera eye also, but what's interesting is it's kind of built backwards from squid eye. In the fish, the eye actually is formed into what we call like an eyeball. And so how that works is it is a ball or a sphere. Check this out. Let me fix this. All right, so we have like a sphere. We think of like an eyeball, more epithelium. What's interesting is the retina is on the inside of the eyeball. So the retina, or the pigment patch and tissues that support the retinol, are on the inside. Retina. And what's even more interesting is that the nerve cells that collect information from the retina are actually on the inside of the eye pocket, the eye cavity. So the nerve is on the inside. This kind of leads to a problem, is that the nerve cells actually have to get out of the eyeball. And so these eyes have what's called I guess the common term for it. So this is our optic nerve. And the optic nerve results in what's called a blind spot. The blind spot is the back of your eye where the optic nerve exits the eye, doesn't have any retina on it. So basically it's a spot that doesn't collect any vision information. Your brain is able to actually cover up the blind spot so you can't really see it, or you don't really see, like, black spots in your vision because your brain covers it up. But there's ways to kind of trick that. If you go to the eye doctor, they'll call this the chiasma. But a chiasma basically is just kind of like a term for a cavern or a hole. So a blind spot is good enough for our purposes. Well, and then we have a lens here. And then there's more epithelium basically that kind of covers the outside here and producing a cornea. But what we have going on here is an eye that superficially is the same type of camera eye. It's also called a camera eye as the squid, but the difference is it's built backwards. That if you were designing an eye, it would seem to make sense to have the light hit the retina before it hits the nerve cell, which isn't photosensitive. So the nerve cells are receiving the message from the retina basically as like a patch that's collecting light. So this one seems to make more sense in terms of collecting light and then sending the message to the nerve cell. In this case, the light actually has to pass through the nerve axons and dendrites and cell bodies before it gets to the retina. And nerves are semi-transparent, but basically it's kind of blocking the light before it hits the retina. And then the retina transfers the message to the nerve cells, which then have to exit the backside of the eye, which causes a blind spot in the back of the eye. So this doesn't really quite make any sense in terms of how it's put together. I guess you really can't argue because, like, this eye is super good at sharp vision. A fish or a shark, for example, can see 20, 30 feet out images, within 20, 30 or more feet. Or if you think of the extreme example, like a hawk can see a mouse at a mile away, so it's like, obviously this has super sharp, super clear distance vision and close vision. So it seems to work really well. But one interesting question is, why is it built backwards? And is this a better design than this is? It's probably that this one ended up evolving sharper vision because the vertebrates are the ones that ended up kind of taking over terrestrial living. Like birds, for example, would have probably the sharpest vision of any animal, and for distance vision. But the real difference here, this is interesting, is kind of where the eye came from. So if we look at the mollusk eye, This is really just a modified version of a simple eye. It's basically where an eye patch migrated back to a brain to send the message to. So in this case, I'm going to say that the eye patch evolved first and the brain evolved later. And that's why the eye here basically is built kind of in the way that makes sense, where the light comes in, hits the retina, activates the nerve, and then goes to the brain. And if you remember from our last exam, that kind of goes along with how the cerebral ganglia basically evolved into a brain here, that the eye patch happened first, and then the brain kind of evolved later to adapt to vision and decision making. It's probably the opposite here. In this case, and embryologically in development, the brain developed first, and then the eye patches evolved from the brain. And if we remember from the lamprey and the shark, I mentioned this diencephalon, which is kind of like this glandular pigment region, the pineal pituitary gland of the brain. Our lamprey basically had a little light patching spot on the brain of diencephalon. The mesencephalon, which is the optic nerve, is right next to the diencephalon. It's likely in vertebrates that vision started at the brain as light-sensing patches that then migrated out to become eyes. So in the case of a vertebrate, the eyeball basically is a result of the brain extending out a photosensing structure from the brain, and which probably results in why it's built backwards. It's kind of like a backwards construction from the mollusk eye. So I would argue that the eye is basically an extension of the brain in vertebrates. And in mollusks, the eye basically is the primary part leading to the brain, or the eye basically evolved back into the brain. And so it's kind of just two ways to achieve the same thing. Which one's better? They're about the same, but obviously I said that this one has much better vision, especially when you consider birds. But this is pretty remarkable in how, distance vision, sharp vision, and color vision that squids can see. And so they're probably about the same. This one probably has an advantage because this is a separate entity, it's muscular, and organisms that have an eyeball can actually move their eyes independently, whereas the eye pocket in the squid was fixed. It was basically stuck to the head, and they couldn't really adjust their eyes independently. But you can imagine here a really good compare and contrast question. Basically go over the evolution of the eyes, and we see the eye spots, the simple eye, the compound eye, basically, and even the camera eye of the squid are all really kind of built the same way. It's just there's slight modifications, namely making a pocket and then making a lens that gives them an advantage. And then the additional question would be on the vertebrate eye. It's basically built backwards, probably because this was an extension of the brain that reached out to the surface of the organism. And so the eyeball basically is kind of like an adaptation of the brain. And the main difference here is that the epithelium is in the back, and the optic nerve is actually in the front of the vision. And that results in this blind spot or the backside of the eyeball. Okay? Questions? Looking good? So I think that would get us, one page question for the test. So you probably want to get a good picture of this. That would be a good way to describe it. And again, the key features we're looking for are kind of the sequence of tissues. Epithelium, retina, optic nerve is kind of the same for all these. And then this one's backwards. Be sure you note that the optic nerve, retina, and then epithelium is kind of the reverse scenario. Okay, so let's go ahead and do like a long 10-minute break. I have a bunch of amphibian stuff, reptile stuff, and bird stuff to look at here. It's not too complicated for that, but there's definitely a lot of things to consider. So let's do 5 minutes. For those of you who came in later, we'll get your test back on Thursday for sure. And then we have a lab exam next Thursday and a lecture exam 2 Thursdays away. So we're wrapping it up here, okay? All right, let's take a long 10 minutes here.